So here we're going to talk about the consequence of mutations. So what I have written is a short mRNA sequence from a gene. Now this mRNA sequence is just somewhere in the middle, so it extends, and remember it would start at the methionine somewhere, and then it would extend on to the right until it hits a stop codon. Now, a mutation is when the DNA in the genome is changed, and if the DNA is changed, when the template is read, the mRNA will be different as well. So this short mRNA codes for threonine, valine, glycine, threonine, valine, proline, serine in this short segment that I have shown here. Now, if there was a mutation in the DNA would cause the change to also be shown in the mRNA. So let's say this GGC here that codes for glycine, if that codon was mutated because the DNA changed such that it was GGU, right? This is what we would call a silent mutation. The reason this is a silent mutation is if we look down here at our genetic code, we see that GGC codes for glycine, but GGU also codes for glycine. So even though we had a change in the DNA that changed our mRNA and changed the codon, because of codon degeneracy, in other words, where there's more than one codon that codes for the same amino acid, this is a silent mutation because the sequence of our protein did not change. And this is the first example of what's called a substitution mutation, where we just swap one nucleotide for a different one. So that's substitution mutations. And we see that the silent mutation usually happens when it's the third nucleotide within a codon that's changed, because that's where we have the most degeneracy. Now, if we take a look at a different example of a substitution mutation, where perhaps this A that is the first nucleotide in the codon of threonine, if that A is changed to a G, this is now going to change that sequence, because ACG codes for threonine, but GCG codes for alanine. Therefore, that change has actually changed the sequence of my protein, because as the ribosome is reading through this, an alanine will be placed there based on the GCG codon instead of threonine. And if this is a sufficient enough change in the right place, that might change the fold and therefore change the function. If the substitution mutation is enough to change the function, we call that a functional change. However, sometimes the new amino acid can be similar enough, maybe they're both nonpolar or they both were acidic and so forth. If they are similar enough, it might not change the fold and the function, and so that would be called a conservative change. It still changes, but maybe doesn't cause a loss of function for that protein. The final possibility with a substitution mutation is what's called a nonsense mutation. And this is where you accidentally make a stop codon. So if we take our example here, and we had a couple of different changes. For example, if our GGC was changed to UGA, UGA now codes for a stop codon. This is a nonsense mutation because once the ribosome got to here, it would now stop, and you're basically missing the rest of that protein. This is obviously a very consequential mutation. It's going to cause major problems for the protein because you're missing the rest of it past this mutation. So we would refer to this as a catastrophic mutation. The nonsense mutation is catastrophic because it is always going to cause major problems with the protein's function and therefore might be a disease-causing mutation. Other examples of catastrophic mutations occur when we have either insertions or deletions. And this is where instead of swapping one nucleotide for another, either a nucleotide gets completely missed during replication or an extra one gets added in. And so let's take, for example, this G here. Let's assume that that G was just missed, so it's lost, it's deleted during a replication at some point. And if that happens, we now still have to bundle our codons into groups of 3, But without that G, the next codon is GCA, those three right there, and then the next one becomes CGG, and then UGC, CUU, and so forth. And what you can hopefully tell is that this is going to cause a change everywhere from there on out. So our ACG obviously is still threonine, GUC is still valine, but now GCA, if we look over here, GCA is alanine, CGG is arginine, UGC is cysteine, and we could keep going on. So if we insert or delete, in this case we deleted, if we delete a nucleotide, that's going to cause everything past that point to change because we've altered the reading frame. We've altered which groups of three, the rest of those proteins behind that insertion, how those groups of three, those codons, are read. And that's going to change the entire protein from there on, and that's why that's catastrophic. It's the exact same thing if we insert a nucleotide as well. If we were to add an extra G in here, so right in this spot here, if there was an extra G inserted in that spot, that is also going to change the reading frame. So now we have our ACG, we have our GUC, we now have GGG is the next three. From there we have CAC, GGU, and so forth. And so you can see how the reading frame from here on out starts to change because, again, we're reading in groups of different groups of three. And so that also, by altering the reading frame, that insertion is a catastrophic change as well. So any insertion or deletion is always going to alter the reading frame as long as if there's only one or two nucleotides added or removed, and that causes major issues. And other than insertion and deletion, we also have the substitution, and there is one catastrophic mutation possibility with substitution, and that's nonsense, where the stop codon is made instead. But even a substitution can cause a change in fold and function if the amino acid that is changed is important enough and therefore alters the fold. But a silent mutation of substitution will never cause a problem because the protein sequence is essentially the exact same thing as it was before.